broad audience, and also to follow Jennifer Mather's presentation, which is quite similar, in fact. So we're talking about variability within behaviour, and I look at coloration patterns. So Hannon and Messenger in particular did some beautiful work back in the 80s where they looked in detail at the coloration patterns of juvenile cuttlefish and how they're controlled, how they're made up of a large number of smaller components I'll show you in a moment. And they suggested at the time that the animals produce about 13 kinds of body patterns, some 5 of which are used, expressed chronically, for camouflage. And subsequently, this idea was, I should say, simplified a little further by Roger Hanlon, who drew attention to 3 main kinds of camouflage pattern, which he called uniform, mottle, and disruptive, and showed that there are similarities between these patterns and the kind of camouflage patterns that other organisms that don't vary their appearance use both on land and in aquatic environments. So the question that interests me is how the animals in fact perceive the world and control then the expression of the different subcomponents in their body patterns to produce, in their appearance to produce these different kinds of body patterns. And rather, as Jennifer was saying, how much variability there is within these patterns. And some people, I don't expect anybody here, think that the marvellous camouflage of cuttlefish is produced essentially by copying the background, that the animals act as some sort of photocopiers. And clearly this isn't the case. What we're looking at is really rather a complex sequence of neural processes, and I have purposely not joined these boxes with arrows, because we're not quite, the arrows tend to go from left to right, but roughly speaking, this is what I have in mind is going on inside the cuttlefish's brain when it observes a background, and that leads to the production of a camouflage pattern. So they start where I sort of work in my real life, in low-level vision, identifying relatively local, simple visual characteristics of the environment. And they use that information then to decide what kind of background in visual environment they're in. And the sort of classification we've emphasised in our work, I don't think it's by any means exhaustive, is a distinction between sand, pebbles, and more three-dimensional environments, gravel, and so on. And these roughly map onto the uniform, mottle, disruptive type of scheme that Roger talked about. And we can then imagine a stage where the animals, perhaps, this is what I'm going to come on to later, have some sort of overall body pattern that they produce. And they produce this body pattern by expressing a number of different components with different weightings. And it would be, for example, very interesting to know whether there's direct connections going from stage 1 to stage 4, from stage 2 to stage 4, and so on. What are the sort of arrows generally running from left to right, weightings and linkings, these different processes, how deterministic they are, how much variability they are. And despite what the ethics committee who looked at our proposal a year or so ago said, which was everything is possibly that could possibly be known about cuttlefish's camouflage has already been studied, and why do you want to do more? I really think actually our knowledge this kind of question, how they use visual information to produce a diversity of patterns, is very poorly understood as a visuomotor system. And what I want to talk about now briefly is just how we're beginning to look at that, or rather how Jay Culligan and Andy Philippides, my colleagues at Sussex, have been doing so. So we collected yet more video, which somebody may or may not want to put on a general use library, Danby. And what Jay did was to film about 10 animals on 6 backgrounds, rather standard sort of aquarium backgrounds, gravel, sand and larger pebbles, and also 3 checkerboards of different size, I think 3mm, 8mm and 12mm, something like that. Checkerboards, juvenile animals. You filmed them for about 30 minutes each and looked at the diversity of what they did. And the way that he did this was a conventional method in our lab, borrowed from Roger Hanlon to some extent, and that is to score in an image the relative level of expression of each of the 35 or so pattern components. And one can do this quite reliably, quite high degree of repeatability, and so on. So it's a pretty robust method. You just look at an image, score these components, and then, a bit like Jennifer Mather, we do a principal components analyses on these image scores. And what that then gives you is, first of all, it sort of washes away some of the variability, observer error, and gives you a relatively low number of principal components in the patterns that account for the main kinds of variability in the behaviour. So we can see here a score actually from some work done by Sarah Zielinski, where red corresponds to a high weighting of a component in a PC, and blue to a low weighting. We can see that these 2 principal components in this case correspond roughly to a disruptive and a Mottle pattern. They don't always come out like that in behavioural in our studies, and quite often it's interesting, one sees 2 patterns that we would classify as disruptive that come out as separate principal components and seem to be more or less as orthogonal degrees of control in a particular experimental situation. Anyway, he subjected scores to PCA from his experiment, from his set of videos, and he then found 3 principal components in this case, described I think about 60% of the variation, pretty typical for these kinds of experiments. And so in a 3-dimensional case, not a 2-dimensional case, as illustrated here, he applied Gaussian mixture models, which essentially attempt to find clusters in the pattern space of the data. And you can then look and ask what these clusters are, where are they centred, and what's the variation within these clusters. That's what we really want to know about. And, whoops. So it turned out there were 6 clusters, which I should immediately say is a bit embarrassing, because we also had 6 backgrounds. And so I cannot be confident that these clusters do not correspond to the backgrounds, but I don't think that they actually do for various reasons. We need to go into that in a bit more detail. Anyway, in this 3-dimensional space, there's 6 basic kinds of patterns, and what we can see in these different views of that 3-dimensional space is there's obviously quite a lot of variation within these patterns. Within these clusters. I should also say that the clusters are quite robust. They were subjected to various kinds of standard control, like dividing the data in half and then doing the cluster analysis on that, Gaussian mixture model on that, and then applying it to the other half of the data and showing that it accounted for the variation within that very well. So we're not just classifying randomness or something of that sort. So the work's done. It's been done quite nicely. And so we can see then there's considerable variation, and we have here a figure from J. Culligan, illustrating each of these basic kind of patterns, and he's chosen, gives some idea of variation. Obviously it's three-dimensional, an image typical of an animal from the centroid, in each place the centre of the cluster, and one far away. And he's given them names, and what we can immediately see is that these names do not correspond in a very simple way to the traditional body patterns represented by Roger Hanlon first, and talked about by Hanlon and Messenger, of course, that I started off with. So the real question in my mind is, are these clusters real, and how much, when we see them, do they depend on the constraints imposed by the kind of environments that we have in aquaria, which tend to be relatively simple in their stuff that you can put on the bottom of aquaria or make with a printer. So again, we go back to Jennifer's point that it would be great to have more field data to understand the variability of these patterns and what they're actually doing, mixing them, whether there are discrete types which are then varied in nature. How much time do I have? Another 5 minutes to talk. That will do very nicely. So, another question we were interested in was when we put the animals on a background, clearly they don't just stick with a fixed pattern. They change their appearance. They move around a bit and they change their appearance. And we knew from earlier work that I'll mention in a moment that in some situations the cuttlefish switch in a very fixed sequence between coloration patterns, so have a sort of deterministic sequence of changes. So we wanted to know, in this camouflage situation, do they have a deterministic set of pattern changes, or is it more random? And where you see a fixed sequence of pattern changes, this comes from the work of, very nice work of Kerry Langridge, also done at Sussex. And what she showed, looking at defensive behaviour, was that as a fish, which is a threat, or indeed another cuttlefish or a crab, approaches the cuttlefish, it goes through a fixed sequence of responses, escalating and then de-escalating the defensive response so deterministically that you can actually read off precisely whether or not the fish, the threat, is approaching or receding from the animal. There's no variation at all. The only variation that occurs really is according to the nature of the threat. So also, as Jennifer mentioned, they produce eye spots in response to visual predators, and actually low-level visual predators, not really dangerous ones, such as small mullet and sea bass, but not to non-visual predators and not to very dangerous threats, which are not going to be scared off by these rather puny eye spots. So highly adaptive and predator-oriented. The main point here is that the behaviour is deterministic. So, having done his cluster analysis and found his 6 clusters that I just referred to, Jay was able to look at the sequence of responses, patterns used by the cuttlefish on his 6 backgrounds. And so what he did was to score transitions between the clusters, and without going into any details, we can see here that he's plotted the directions and the frequency of the transitions in the data, and given that the animals express different patterns with different probabilities on different backgrounds, which they clearly do, the way in which they change between these patterns is completely random. And it's both random in the first order and the second order. There's no statistical structure here whatsoever. So that's nice and simple. And in one case they're behaving completely deterministically, it seems. In another case they're behaving completely randomly. And why they should do this is an interesting question. And I think that's where I'll leave it.